It is my absolute privilege to introduce our keynote speaker. By the way, I love that music. Thank you. What I'm about to share is a shortened version of her bio, but a longer one can be found on the CASSS website, which I encourage you to read. Dr. Sonia Vallabh co-leads the initiative to develop preventative drugs for prion disease at the Broad Institute. She comes to this work with a personal mission, which I will let her share with you in a few minutes. At the Broad, Dr. Vallabh is focused on developing preventative drugs for prion disease. Lowering a prion protein, PRP, is a genetically well-validated strategy for delaying the onset of prion disease, and it lends itself to the measurement of PRP as a biomarker available before the disease process has begun. Dr. Vallabh holds a BA from Swarthmore College, a JD from Harvard Law School, and a PhD in biological and biomedical sciences from Harvard University. She trained in Stuart Schreiber's laboratory at the Broad, and in 2016, she served as a patient representative in the White House Precision Medicine Initiative. Please join me in welcoming Dr. Sonia Vallabh.
Hi, everyone. It's really my pleasure to be here with you to get to share the stage with people like Jamie and Kim and Dr. Shapiro. Congratulations. And I'm excited to get to tell you about what we're doing at the Broad. As Kim alluded to, this is a personal mission for me, so I'll take you back to the beginning. These are photos from my wedding 15 years ago. That's my husband, Eric, and I, and that's my mom helping me get ready that day. So she actually, I can't take any credit, she organized the whole party. She was 51 years old and healthy. It was a beautiful day. And what we couldn't have known at the time is that this was the last family event for which she was with us. The next year in my family's life looked like this. So from completely healthy, within the range of what's normal for any of us, in January, my mom started to experience kind of a miscellaneous group of symptoms, changes in eyesight, rapid weight loss in February. And what I want to emphasize here is that by March, she was not fully with us. She could not finish a sentence, and she didn't know what was going on. What were we seeing? So I just have to emphasize to you, right, that I'm telling this story like a thing that happened, and it seems a little bit inevitable now. But at the time, it was completely unclear what was happening. And to see someone decline like that, it's almost like a time-lapse video of a person aging and dying, was so surreal that it seemed to me, very naively, surely we would put her in front of a group of smart doctors and they would say, this is X. At a minimum, this horrifying thing should have a name, right? But actually, we took her around all year to fancy places, right? Like the Cleveland Clinic and Brigham and Women's Hospital. And she nevertheless died without a diagnosis at the end of that year. And it was only from her autopsy that we learned that she had died of genetic prion disease. This was loaded information. This is how we learned that her disease had been genetic, and this is autosomal dominant disease, and I was at 50-50 risk of having inherited the causal mutation. Eric and I decided almost instantaneously that it was important to get predictive genetic testing, which we did. And it was the end of 2011 when we got this genetic test report on the left. And it wasn't good news. So I did inherit the mutation from my mom. At the time, Eric was trained as a transportation engineer and I was trained as a lawyer. So we were in very different worlds. We were not biomedical people. But this was kind of explosive information. And we ended up feeling highly motivated by the experience we had just gone through to educate ourselves about what we were up against, because it very much seemed to us that doctors didn't know what this was, and that although then as now prion disease is fatal and untreatable, if any progress was made over the next however many years, we were going to be responsible for knowing that and for following that and for being savvy consumers of scientific information. So this is how we took up sort of educating ourselves about prion disease as a hobby, but it acquired a lot of momentum fast. We ended up leaving our jobs for entry-level jobs in biomedicine, and from there, starting a nonprofit, beginning our PhDs. We were super, super lucky in that process to find our scientific home at the Broad Institute, Cambridge, Massachusetts, and we're there to this day. Now we run a lab of 14 people, and we are ultra mission-oriented around getting a therapy to people in our lifetimes. It's a privilege. It's a really good group, and that's what we do. So I'll tell you a bit about what that looks like on the ground. The mission is our North Star, right? It never wavers, but what we need to do to get there is constantly enlarging. It's constantly surprising us. And I'll kick us off by just telling you a bit about the disease. Anybody who sees prion disease clinically is familiar with this alphabet soup of like CJD, FFI. There's a lot of names. Famously, there are 3 different etiologies. Most cases are sporadic, by which we mean random. No known genetic or environmental trigger. About 15% of cases are genetic, people in the position that I'm in. Vanishingly rarely these days is prion disease acquired, but of course that's what made prion disease famous. Early symptoms, they can be almost anything, but they very rapidly converge towards a profound dementia, a profound and rapidly progressive dementia. Despite all of this variability on the surface, prion disease has a unifying and really simple molecular blueprint, and this is our greatest asset. I return to this every day. Single gene, single RNA, single protein. That single protein has this unique ability to misfold into a conformer, now called a prion, that can go around grabbing normal copies of PRP and misfolding those. This is how you get a sort of domino effect of prion formation through the brain that kills neurons. Our lab for years has been focused on the therapeutic hypothesis of lowering the amount of normal prion protein in the brain. Why? We were the lucky heirs to a bunch of information, right? We came along after the originally controversial prion hypothesis had already been well accepted by the field, and a whole bunch of foundational experiments had been done. What you're looking at here are data from prion-infected mice. So we literally can model this disease by injecting prions into the brain of a wild-type animal, and they go on to develop full-blown, terminal, faithful prion disease in a stereotypical amount of time. So disease of mammalia, right? These are not really models. This is just prion disease. If you do that in a wild-type animal with normal PRP levels, they get sick, if it's a mouse, in 5 months. If you overexpress the amount of prion protein in that animal's brain, it gets sick faster. If you knock out 1 copy of the gene, it takes more than twice as long to get sick. If you knock out both copies of the gene, this animal is now invincible. You can put billions of infectious units into its brain, and there just is no template. There's no fuel to the fire, and the disease can't happen. The next question is always, and rightly so, would it be safe to reduce the amount of normal prion protein in the brain? And gosh, do we have a lot of lines of evidence that say yes, including 8 different knockout mouse lines at this point, knockout cows and goats and rats and hamsters and heterozygous loss-of-function humans, and genomic analyses across population data sets showing us that natural selection does not mind if prion protein gets knocked out by accident. So, with all of that said, I think we have this therapeutic, strategy, we have this protein that is essential for disease, expendable for healthy life, based on everything we know today. And the question becomes, what do we do about it? How do we go into the brain and lower the amount of this protein? Super, super clear mandate. To summarize 10 years of work in one slide, the first therapeutic strategy we started working on was antisense oligonucleotides in partnership with Ionis Pharmaceuticals. And that top plot is kind of the headliner here. In the blue dashes, you're seeing animals that started getting ASO treatment. So these are oligonucleotides that bind the prion protein RNA and trigger it for degradation, so less protein is made in the first place. And we think that these ASOs are reducing prion protein levels in the mouse brain by about 50%. If we start dosing early, right, so these animals are getting chronic dosing starting before pathology and before symptoms. They are redosed every 90 days. In that paradigm, the gray line is the animals that got no treatment. They die very predictably. The long blue dashes are the animals that got that chronic therapy. The red dashes are the animals who had one copy of the gene knocked out genetically. So we can basically replicate the benefit of knocking out one copy of the gene with chronic ASO therapy that starts early. In the plots below, I'm showing you what happens if we treat the animals after symptoms. On the left, this is a cohort of animals that already looks sick. Like, I can look into the cage and say, they've lost weight, they are scruffy, they are not grooming properly, they are not building nests. If we treat at that time point, we can extend survival for maybe a third of the animals. They don't lose their symptoms, they don't recover weight, but they survive longer in a disease state, some of them. On the right, this is a cohort of animals where we let them go all the way until some animals in the cohort were terminal, and then we treat the rest. So this is advanced stage treatment, and it is too little too late. So what do we learn from that? I think that prion protein lowering works, and that the details, particularly the details of when we treat, really, really matter. So I think of ASOs now as sort of our foundation. They're our therapeutic proof of concept. They have allowed us to show pharmacologically what we already knew genetically. And they've been sort of a motivating force on top of which we've built a lot of infrastructure. So I won't bore you with all the details of these things, but we have, the natural history, the pharmacodynamic biomarker work now, the penetrance of PRNP variants, right? We have been trying to figure out, in light of sort of like constant recognition of our enlarging mission, not just what could the molecule be, but what are all the supportive pieces that we need to put together if we're going to do meaningful clinical trials in this disease. And especially around that sort of timing question, I want to talk more about what the right trials might look like. So we have two patient populations here. And I told you that symptomatic patients die ultra rapidly. Less than 6 months on average from first symptom to death. We can almost think of this as a lightning strike. There are precedents for doing trials in this population, and you can really rapidly get a survival readout from those trials. What is the highest good you can do? Realistically, based on everything I've seen, the highest good you can do is to stabilize, right? If you lowered PRP to 0, which we're not capable of, maybe you could stabilize a patient, not in the state that they were at when you first saw them, right? Because time passes. You have to randomize them, you have to enroll them, you have to do all the paperwork, they have to have their baseline visit, okay? Not in the state that they are when they get their first dose, because the drug has time to effect, and the native protein has a half-life of 5 days. Probably the highest good we can hope to do for a symptomatic patient with a perfect drug is to stabilize them in the state where they are a month after their first dose. This disease moves really, really fast. So I really hope we are going to be able to do meaningful therapeutic benefit for symptomatic patients. And I also want to be honest about what an uphill battle that is. Our second population is pre-symptomatic people. With predictive genetic testing, we can identify people at risk, like myself, years or decades in advance. We know the high penetrance variants that basically tell people this will almost certainly happen to you sometime in adulthood. There are low thousands of such people in the US. If we want to think about doing a pre-symptomatic trial, which, by the way, there's no precedent for this because this just isn't how we think about drug development traditionally, what can we measure? These people are healthy, right? They don't have a phenotype. And age of onset is really variable. So to give you some sense of that, average age of onset, median age of onset for my mutation is 52. Standard deviation is plus or minus 12 years. And we've seen a case as young as age 12. Parents' age of onset, sex, etc., these things do not predict when someone will experience onset. Literally, this can happen anytime. So when you combine rarity and variability in age of onset, what you get is a lack of power to recruit people in a healthy state and then randomize them to onset in a pre-approval format. We don't have the numbers for that. I also don't love the idea of recruiting, the carrier community. They're my people, they're my friends, and I don't love the idea of recruiting them and randomizing them to the death. I don't think you would love it either. Okay, so if we can't do that, what can we do? I think trials in carriers are going to rely on a biomarker endpoint. And we can imagine different kinds of biomarkers that could be. And to start pushing on this, we started enrolling a natural history study in 2017 at Mass General Hospital. These are carriers who are coming to Boston. They are donating a couple days a year. Their spinal fluid, their plasma, they're doing a bunch of tests, right? They're taking time off work for this. And our retention rate is amazing. This is a really, really motivated group of people. And our first report out of this study in 2020 had what snobby academics would call a negative result, how disappointing, but what I thought was really fabulous as sort of the top line result, which is when we look at spinal fluid and blood in these carriers, they are indistinguishable from healthy controls. So this is not like a Huntington's disease, genetic Alzheimer's, FTD type story, where you might be used to the idea that molecular biomarkers and brain changes are happening 10 or 20 years in advance of first symptoms. Instead, it actually looks like people are healthy for some variable number of decades of their life. And then when things start changing, that happens very rapidly. 4 years later, we published a second report. Now we've seen 4 conversions to symptomatic disease. By the way, this has been heartbreaking for the study team. But this is what happens, right, when you follow people longitudinally. And here are some, usual suspect type neurodegenerative disease biomarkers, markers of neuronal damage, of reactive gliosis. These things are off the charts in symptomatic patients because the brain is dying so quickly. But when we look at our carriers, again, normal for years. And is there an uptick in this stuff that predicts onset? Meh, not really. Really, the changes happen at onset. And anything before that is unreliable and very brief. Pivoting gears a little bit, here's a fundamentally different kind of biomarker. We're no longer looking at pathology now. We're just looking at the normal prion protein, which, by the way, you all have. You all have it, right? In every tissue of your body, most highly expressed in the brain. And when we measure CSF PRP levels, this is not about disease process. This is about baseline state. These levels, provided that we do all the things right, you collect your samples right, you store them right, whatever. If you do that, baseline, CSF PRP baseline, is really stable in an individual over years. I think this is great news because if our goal is to lower PRP in the brain, CSF is our best shot at reading that out. And what we see here is a stable baseline on top of which we should be able to really reliably measure a drug-dependent decrease. In support of that, we've done ASO treatment in rats, and measured sort of dose dependently that if we lower RNA in the brain, we lower protein in the brain. And if we lower protein in the brain, we lower protein in the CSF. So we'll keep doing this, right? We'll keep doing natural history. But I think what we can say so far is carriers are healthy, by and large, cross-sectionally. We do not have the tools to predict onset. You know, it's coming in 6 months, it's coming in 2 years. CSF PRP behaves really well, and we can count on it. And let's not forget, right, that when we treated our mice, we saw the most beautiful result when we treated before pathology ever began. This is common sense, right? Like, none of us are surprised by this. So when I put all of that together, what I see is a mandate for primary prevention. What we need to do for carriers is get to them before the pathology does. Like, how insane would it be to be me, right, to be in my position, to have a genetic test report for years and years and years, to sit on it and do nothing, and then to wait until a lightning strike disease is killing your neurons literally by the minute, and then try to do something about it. Like, that is not the highest good we can do, right? Okay, so we've had conversations about this. How have they gone? You guys don't need background on accelerated approval, but basically, if you want to be leaning on a biomarker as your surrogate endpoint, it needs to be reasonably likely to predict clinical benefit. This is the pathway you go through. Your analogy is viral load in HIV. I think we have a really strong analogy here. This protein is our pathogen. Of course, you still have to show safety, you still have to show efficacy, and you have to show in post-approval format that your biomarker did predict clinical benefit. So make good plans, do all these things. When I look at what's required to sort of qualify for accelerated approval according to Congress, it kind of feels like this was written for us. The strategy ends up looking a little something like this. You know, it's not specific to ASOs, it's not specific to an RNA targeting drug, it's just anything that lowers PRP in the brain. Put it into carriers, read out CSF PRP levels, see if they go down, and if yes, let's call that a primary endpoint reasonably likely to yield clinical benefit, to delay or prevent prion disease in these people. So we've been talking to FDA about this since 2017. I was really moved by our first meeting because I will say, without, wanting to throw anybody under the bus here, but nearly everyone we talked to about this meeting before it happened, either in neurology or in pharmaceutical space, said, "You're crazy, they'll never go for that." Like, why would you waste your time traveling down to DC to pitch treating perfectly healthy people to prevent a disease? That's not how it's done. And then we went into this meeting and we're told almost right away, you really don't have to rehash your case or your materials that you sent. We've reviewed it all and we get it. This is the disease you need to prevent. Let's talk about what you need to do next. And there were things on that list, and we're doing those things, right? One of the things was, let's nail the therapeutic hypothesis to the ground. Let's be so sure that prion protein is causal in this disease. And so we've gone out since then, and we've shown if you lower PRP, that's helpful not just in mice, right? But in mice infected with 5 different mouse-adapted prion strains, and in hamsters, and in rats, and in humanized mice, and in spontaneous models of prion disease. Good. Let's be as sure as we can be. So yes, we left with homework, but we also left with a signal that this is a path that we can walk. So what comes next? All that ASO work led to the development of Ion 717, which is now clinical stage. It reached first in human at the end of last year, sponsored by Ionis Pharmaceuticals, and now that first in human trial has fully enrolled. 56 people are part of that trial, and maybe we will get some top-line result by summer. This trial, in early symptomatic patients. I hold open a lot of caution, right, and a lot of possibility for what we will learn from this trial. We've already learned a lot. It enrolled super quickly. just like I said it would. This is a super motivated community. Have they caught people early enough? Are we getting good enough brain distribution, because we are a whole brain disease, by bolus intrathecal dosing 2' MOGAPmer ASOs? I don't know. These are the things that hopefully we'll be learning soon. We support, that work in every way we can. And in addition, here's what is the beating heart of our lab right now. We need next and better drugs, right? Better biodistribution, better tolerability, better potency, better durability. We can do better on every dimension. The technology exists, right? It exists on Earth. If we had come to this 10 or 20 years ago, I wouldn't be able to say that. And we need to continue to fight for prevention. So I'm going to talk quickly about 2 other strategies that are live in our lab right now for reducing prion protein. One is sort of a next generation oligo strategy. So this is an siRNA duplex, but 2 of them, linked with a PEG linker, technology developed by Anastasia Kurova at UMass. For whatever reason, this higher molecular weight entity distributes better across the brain, hangs around longer than a traditional ASO or siRNA. The other strategy is a little more out there. So this is a gene therapy, but really it is an epigenetic editor called CHARM. This is developed in collaboration with Jonathan Weissman's lab at the Whitehead. And the way it works is basically by installing permanent methylation at the PRNP promoter. So go right to the source, tell the gene to shut down, not make any RNA or protein. The thing about these drug development programs is that neither has a commercial sponsor. The sponsor is us. We're working to develop these out of our lab and take them forward into small investigator-initiated first-in-human clinical studies. So that's the whole thing. I'll tell you a little more about it. Okay, here's a bit more about the divalent siRNA. Just to say, this is actually, at this point, quite a mature program. We've done GMP manufacturing and GLP toxicology in small and large animals. The data look clean, and the goal, my husband Eric isn't here right now, and where he is actually, he's hunched over his desk, feverishly writing the IND, which he's almost kind of doing by himself. That's not that much of an exaggeration, right? There's a ton of CROs that have contributed reports, and there's a handful of consultants who are helping him quite a bit, but anyone who's touched an IND or seen one from any distance knows that this isn't actually a one-person job. So that's been hard to watch, honestly, but it kind of seems like what we have to do. He's hoping to submit next week. These are the ways that maybe a divalent siRNA will differentiate from an ASO. We won't know until we try it, right? We won't know until we try it. Even if it performs beautifully, it will still be an intrathecally dosed, lumbar puncture delivered drug. We will still have a gradient, right? We will still have to get people to a tertiary care center where they can be LP'd on some regular basis. So there are drawbacks to this. But there's a race to the first drug, there's a race to the best drug, there's everything in between, right? This is what progress looks like. Here's a bit more about CHARM. So as I mentioned, what a more complex drug type, right? And Eric keeps saying, will you stop pretending like oligos are simple? But ever since I started thinking about gene therapy, it's a whole nother level. But basically the goal is you get this molecular machine to the gene of interest, to the promoter, and recruit an endogenous methyltransferase that stamps that gene with methyl marks and says shut down. And the knockdown that we've seen using this strategy, best I've ever seen in my life. We're talking about, in whole brain hemisphere, 80% reduction of prion protein. So it's stunning. And here's some requisite, really beautiful images to show you that, PRP positive neurons in yellow go almost completely pink, right? Almost completely PRP negative with therapy. 5 years ago, I would have said, I can't afford to spend any time on gene therapy. It is not realistic for a whole brain disease. I am, humbled and grateful to have had my mind changed about that. So first there was PHP-EB, which some of you may be familiar with, a viral vector capable of transducing neurons, most of them in mice, following systemic dosing. Then PHP-EB failed to translate to humans, and it was a big disappointment. Then the same person who developed PHP-EB, my friend Ben, shown here, came back with a receptor-first screening approach and developed vectors that bind the human transferrin receptor. What do we know so far? That you can deliver them systemically in a transferrin humanized mouse, and they light up the brain in a beautiful way. So, I got through all that in order to show you guys all that. What I actually want is a chance to talk to you, and I'm going to take that chance now. This has been a real journey, and I think developing these drugs ourselves has taught us a lot, maybe a lot of stuff we never wanted to know. It's been really interesting kind of butting our heads against a system that is set up for commercial sponsors, a system that wants to do things at a certain scale, at a certain price point, and at a certain speed. And we have spent, months and months and months of effort. We basically have a full-time person on our team whose job is call people over and over and over and over until you find someone who is willing to be a little bit more flexible and creative about taking on a small project. We're not one size fits all. We're a really, really different size. And that, even just that itself, is a big struggle. I think, well, what I wrote here is anyone other than us would have given up by now. I actually feel that every day. I feel that we are sort of like, solipsistically, a fulcrum on which the world turns because everybody else has the luxury to say, gosh, this got really hard. I don't like this anymore, right? Like the science was fun, like directing the experiments in the lab, doing them myself, being hands-on, gathering the data, that moved fast, that was awesome. We've gotten to a part in the process that doesn't feel fast or awesome. So I'm gonna pivot to a different disease and do that discovery work again. We don't have that luxury, right? We're like in this for the long haul. Here's another controversial reflection. You wouldn't believe how many people say to me, Sonia, just use AAV9 because there's a streamlined regulatory path. You can reference other submissions. There's a lot of NHP data on AAV9 safety. You could move so much faster. As if moving fast is the highest and only good. As if it's only secondary whether the drug works. Because AAV9 is not going to get enough neurons in the adult human brain. I haven't seen any data to convince me that it would help patients at all. But that almost gets backburnered, right? People have really pushed against us wanting to move forward with a novel vector that has a shot of reaching all the neurons in the adult human brain by systemic administration. And I really have to push back on them in return. I see the same thing in my friends who are developing N-of-1 oligos. They're so wedded now to 2' MOGAPmer chemistry because there's an N-of-1 guidance. And they say, oh, if we do it this way, we can maybe get there in 18 months. But that way is no longer the best way. And I just this really painful pull towards doing things the way they've been done in the past because it's faster. It's a tension that I am struggling with. It's inevitable, I think, but how to be mindful about that and push against it because the pace at which these technologies are improving is so exciting, right? And I don't want to be the person, right? I'm the front person for my community. I don't want to be the person saying, sorry guys, I'm not even going to dream of getting the best thing for you. Okay, let's talk about monkeys. So Ben Deverman's viral vector that binds the human transferrin receptor is human-specific. It doesn't bind, because of a single amino acid difference, it doesn't bind NHP transferrin receptor or transferrin receptor in any other species. It will bind in humanized mice, it'll bind in humans. And you wouldn't believe how many people have said to me, well, okay, it's dead in the water then. Let's not try it. These are patients, my patients, they're gonna be dead in 2, 3, 4, 5 months. And when I think about, let's say this vector was cross-reactive with NHP. Let's picture what that study would cost. It would cost us probably 2 or 3 years. And maybe you're saying, hey, no, I think for this disease you could do 3-month in life NHP study. You could do a 9-month in life. That's not 3 years. But I've come to appreciate that it is. You spend all that time contracting. You spend all that time finding those few existing NHP slots and sitting in line waiting for them. And then there's the in-life portion. And then there's all the formal reporting out, right? Because this is GLP, and it's going to take us ages to get you the interim report and the final report. So really, we are talking about 2 or 3 years. In those 2 or 3 years, 1,500 people died of this disease in the US. Most of them wrote to me and said, can we please try an experimental therapy? And I had to be the person to say, no. It's complicated, right? Around at what stage should people be able to take risks and define their autonomy in this way. But these are people who are on death's door saying, I want to do this, even though I'm almost certain it's not going to help me. I want to do this because I want this to mean something. And their families are saying that too. And prion disease, I don't want to pretend like we're the only awful disease. Anyone who has been through a lightning-fast, life-shattering disease, right? It totally busts up the meaning-making process, which is such an important part of grief, right? And the families who come out of this, it's going to take them years or the rest of their lives to make meaning out of what happened and to recover from it. And they are coming to us saying, give me an opportunity to make a little bit of meaning out of this just inconceivably brutal experience that has been thrust upon us. And we're saying, sorry, we need to protect you. We need to protect you from the opportunity to try to make that meaning. This puts me in a really hard position, right? And when I got into research and thought, I want to develop a drug for this disease, I didn't anticipate that that was going to be my job. Okay, final point here. We famously don't have any drugs for neurodegeneration, and it's a really universally sad situation. And what I see is that we're putting billions and billions and billions of dollars into doing enormous trials where we come out and kind of didn't learn anything because people are just wondering, did we treat too late? So people call me radical. They're like, oh, wait, you want to treat before symptoms? That's so out there. But somehow we don't consider this out there, right? To spend all this money on experiments from which we can't learn. So that is some of what I think about every day. What I'll say about primary prevention is, if not now, when? I can't imagine a clearer case for this than prion disease. And the moment is now, right? We have the technology. Okay, a few closing thoughts. Taking us back to the timeline of my mom's illness, my reflection on this, in retrospect, is that there's no moment in this disease course where I would have chosen to be kept alive on a life-extending symptomatic therapy. What would that mean for me and my family? It would mean arresting me at some point on the rapid slide into dementia, and that my family would then be anchored in everything they do by caregiving for me, right? And also, their memories, right? I have a 7-year-old and a 5-year-old born through IVF PGD. Their memories of me would become eclipsed by their memories of my illness. Everybody sees this differently, and I can't speak for other people, but when I look at this, that's what I see. For me, it's prevention or get out of town. This is my final slide. So these are survival curves, sort of like annual probability of onset as relates to age for the 3 most common high penetrance PRNP pathogenic variants. Mine is the one in orange. I turned 40 last March, and in 12% of universes, we've done all this, and we weren't in time, and I'm already dead. That number's only going to keep going up. The day after my 40th birthday, I walked into a conference room at Broad, and we had just received a bunch of CSF samples from genetic prion disease cases. And up on the screen in this room is the demographic information that corresponds to each of the samples. These were samples of people who died from my exact subtype with my mutation. And one of the columns in the spreadsheet is age of death. Virtually every number in that column started with a 4 or a 5. So, was I that much older than a week ago when I was 39? No, but that was still symbolic and a bit heavy. When I think about where we're headed, I think things get a lot worse for patients before they get better. The reality of our situation now is that it's a lot harder to tell people there's nothing we can do for you when I know, and they know, that there's technology on Earth that might be able to help. It's also gotten harder in the sense that when we first got into prion disease research, I very much conceived of this as man versus nature, right? Which is a beautiful thing about science. There's no one on the other side of this, right? No one is rooting for prion disease. And yet, now that we have a therapeutic hypothesis that nothing is bulletproof in biology, but I think is worth betting on, now that we have proof of concept, increasingly, we are in this domain of our quest that is more man versus man. And that to me is so surreal because it's still the case that there's no one on the other side. And yet our greatest sources of jeopardy are human systems. And I think for us, the big challenge, right, is to tap into people's intuition as individuals, into their sort of their common sense and desire to do good as individuals, and figure out how do we get that to percolate upward into our systems. I think that I could talk to you guys all day, but that I should stop there and invite Kim on stage. And thank you so much for your attention.
Well, thank you so much. So now we have some time for some questions. We have some folks that have already put questions in through the portal, but also we invite folks here in the room, if they're interested, to use the microphones to ask questions. Sonia, I'm sure many of us, as I have been, very moved by the human story of this, and then the scientific one. And as you heard this morning, I think this community kind of exemplifies both of those things. But I was curious, as you think about this community, who primarily focuses on sort of the making, the testing, the delivery of the products, if you could share a little bit about maybe some of the the paths that you've taken there, maybe some learnings, some questions that you might be looking to solve.
Sure. Yeah, I mean, I guess I mentioned the sort of challenges that we've faced being small, unconventional sponsors. It does feel like we're swimming upstream every step of the way. One thing we had to go talk to FDA about is the fact that if we conformed to standard requirements for set-asides from our manufacturing batch, that would consume 90% plus of our drug product because we're not making that much. And we just had to be willing to go and ask, can we retain less? Can we shrink down the amount that we're saving for stability and all of these other things? Can we have, vial fill at different levels because that's going to help us conserve volume? I think one thing that we've learned being us is that we have an uncountable number of disadvantages compared to a commercial sponsor, but we have this one advantage, which is that we're willing to ask. And I think that's our only superpower, is that we're willing to ask. And sometimes the answer is no, right? We asked if we could do rodent-only toxicology for our divalent siRNA project, and the answer was no. But sometimes the answer is yes. So, yeah, I don't know if that answers your question, but yeah.
Yeah, thank you. And Andrew?
Thank you, Kim. Well, I would like to thank you very much for this inspirational presentation, and also come here to share with us on your personal story and journey in this, carry this potential disease. Actually, another comment I want to make is this is another example to how important to get a patient's voice into the drug development process, including clinical trials and approval process. So my question to you is that maybe I missed it from your presentation. What is the normal function of prion proteins in body that is non-function if we move into totally knockout of this protein, or what is the current knowledge on that? Thank you very much.
Yeah, fantastic question. So, as with any protein that's been studied for a bunch of years, if you look in the literature, and unfortunately we have no mechanism for cleansing the literature, right? We only accumulate. So you'll find hundreds of hypotheses that haven't replicated. But the one that I believe, because we've seen it replicate in a bunch of systems, is that there appears to be a mild, age-dependent peripheral neuropathy in full knockout animals. We don't see it in heterozygotes, but in full knockout animals, if you look at their sciatic nerve at, mouse that's 60 weeks of age, under a microscope, you can see sort of an onion bulb formation of myelin that's been damaged and repaired. Okay, does that matter? It really depends on who you ask. If you ask a pathologist, they'll be like, oh my God, the onion bulb. If you look at the mice, they're perfectly happy. They look completely normal. If you try your darndest to tease out a phenotype, on some genetic backgrounds, but not others, you can measure a slight difference in grip strength and in latency to jump off a hot plate. So I think this is a thing to monitor, certainly. Would I call this a real concern about knocking down prion protein? I would not. I think one thing we don't know yet is minimum threshold to rescue that phenotype. We know that 50% PRP, you don't see it. We're actually working on generating hypomorphic mice right now to ask 5%, 10%, are these levels enough? Because I guess one thing to bear in mind is that we do not have any tool right now or any prospect of developing a tool that will reduce prion protein in the brain by 100%. So I think there's also just a real therapeutic window question there. But overall, I would say that this is what we know right now about native function. I find it unconcerning. We'll keep looking, we'll keep learning. It's an active area of research for our lab. That's where we're at.
Thank you very much. I forgot my name, Andrew Chen from Novo Nordisk.
Great to meet you.
Thank you, Andrew. One of the questions from online, is there a common symptom for prion disease that could be confused with symptoms of other diseases?
Yes. So here's a challenge. We're a whole brain disease, and I think by the time you're measuring prions anywhere in the brain, the prions are everywhere. And this is reflected in the early symptomatology. The first symptoms can kind of be anything. They can be motor, they can be psychiatric, they can be cognitive, they can be autonomic. They very rapidly converge towards dementia, but part of what we need to do to meaningfully treat symptomatic patients is catch them at the earliest possible moment. And that's hard to do when you don't have sort of stereotypical early symptoms. What I tell neurologists, and I almost never turn down an opportunity to give grand rounds and just beg people to order our molecular diagnostics the second they suspect anything. We have now a test called RT-Quick that is pre-mortem. You seed the assay with a little bit of CSF. If and only if there's a prion in that sample, you will get a fibrillization reaction and a fluorescent readout. This is 95% sensitive for sporadic prion disease and almost 100% specific. It's a great test. It's not as good for genetic subtypes, so in parallel, take a little bit of blood, do Sanger sequencing on the prion protein gene. You put those two things together, and we could catch people with really high confidence. It's just a question of getting the disease profile to a point where people sort of reflexively order this testing. So early symptoms. Here's another thing I'll say is when we think about the comparative advantages and disadvantages of trials in our two populations, symptomatic patients are the gold standard, right? Who feels or functions better or lives longer. But wow, is that a messy experiment? You've got all of these different symptoms, you've got varying progression rate, there's a lot of clinical heterogeneity. When we think about our carrier population, we have a uniformly healthy population. We're going to see safety signals. We're going to have unconfounded access to target engagement data, because in symptomatic patients, CSF PRP is more complicated. In healthy carriers, it is not complicated. So we will get very clear readouts of these sort of top line, considerations. And it is a very, very clear numeric readout whether we've lowered PRP. So our primary endpoint becomes this thing that you can just measure with ELISA and get a numeric answer out in a few months. So my my perfect trial, sure, we can randomize people. I think we could talk about crossover too, but okay, let's say we're going to recruit carriers and randomize them, but we're going to randomize them for 6 months, right? Or we'll do a delayed start for people who initially get placebo. And basically, you can measure baseline CSF PRP 2 or 3 times. You get that flat baseline, then you dose, then you measure, and you can sort of measure your delta. And you can do all that in a time frame where you don't expect any conversions. So you don't think that you're actually randomizing people to the death. If it's working, you get them all on drug.
Thank you. The question in the back, please, followed by Kavita. Thank you.
Hello, my name is Lindsay Silva. I'm from Genentech. I noticed that the administration is intrathecal and may require high doses. So given the need for having tight microbial control and endotoxin specifications for this administration, have you had any challenges in terms of making sure the method is sensitive to detect at that low level, and were specifications so tight that you may have had challenges in making the final drug product?
Yes. I'm so glad you asked. Some of this stuff, I block it, but now I'm remembering. So when we went to manufacture the divalent siRNA, right, I haven't even told you guys how we scare up the funds to do this stuff, but it is scrappy. It is by our bootstraps, right? So we have fundraised from families the money to do the manufacturing. And the fill finish, I didn't even know the noun phrase fill finish, but as we get into this, these chapters just appear out of nowhere, like, oh, and you have to do X, oh, and you have to do X. And suddenly we've gone from something that I think is going to be a relationship with one CRO or CDMO, and now it's 8 or 12 or 15. And we're talking about sort of filling the vials, and what we learn is that a commercial sponsor would just pay to fill identical vials with, low endotoxin diluent. They would pay as much as you pay to fill your vials of drug, right? And then they would have these identical vials that the clinician really likes because they want to be blinded and they don't know is it drug or placebo, and that whole thing gets stored at yet another contracted facility, right? And then sent to the pharmacy at the hospital. Okay, so I'm coming to understand all of this. We have the money to fill the drug. We do not have money to pay for our own, diluent bottles. So, okay, let's buy low endotoxin saline, right? Let's just buy a product off the shelf. This turned out to be more difficult than I could have imagined. And part of the problem, it was actually just figuring out the diluent that the drug was going to be in, right? Because that's what we want the placebo bottles to be. There was one product on the market where they could guarantee low enough endotoxin, and that product had a little bit of phosphate in it. And the inventor of our drug type, right, had been very, very clear, don't put it near phosphate. It'll be great, but don't put it near phosphate. And we're we're between a rock and a hard place. What can we do? So we embarked on this stability study using this one commercial diluent and our drug, and just got lucky that it was low enough phosphate levels that the drug didn't crash out and appeared to be behaving fine. If we had gotten unlucky, I don't know what we would have done. So there's no one at the helm making sure that all this stuff makes sense and is possible, Yeah, one example. I'm sure there's more.
Thank you for your presentation.
Yeah.
Yeah, thank you, and a very relevant description. Thank you so much. Kavita, please.
So Kavita Iyer from Teva. So 3 things that popped in my mind as I was listening through your talk is, first is gratitude for the current moment. Then the other thing was resilience from your personal story and your journey. And the third piece was scientific curiosity, which is keeping you going despite the challenges that you have faced and continue to face living in the reality. The prion protein really jogged a memory because I was really baffled when I heard, because I started thinking, I have heard this word before. It took me 3 decades back when I first started my PhD career, and there was this one professor in our lab who was kind of nerdy. Nobody spoke to him, but he was known as the Professor Prion because he was the only one who talked about it. So now fast forward, I'm wondering, 3 decades later, we are still here talking about prion, which is still an academic pursuit, and there's not much been a therapeutic solution.
Yeah.
So from that standpoint, from your experience, I'm really, really curious to understand what are the challenges and why has the needle not moved much? It might be a very loaded question.
Yeah.
And as a scientific community here, there's so many experts, so many curious, and Margie's her living example, how much she has done to the community from a regulatory standpoint.
Yeah.
What could we do to really help such situations get to a better state?
Yeah, thank you for those questions. Why has the needle not moved? First of all, whoever your prion guy is, I probably know him, so I find that hilarious. Of course, no one talked to him. But why has the needle not moved? I think the most generous answer is that the technological moment was not ripe. There's a lot of other reasons, and there's a lot I could say about academia and its flawed incentive structure, but you guys know all that stuff. But I actually do think, 2014, the moment when we were beginning our PhD, was the moment that Ionis published that bolus dosing of ASOs into CSF could achieve some biodistribution into all brain regions, right? Not perfect, but some. And that was the first thing that made us think an oligo drug could be a path forward here. I think 10 years prior, people were thinking on some level about prion therapeutics. There were still some tough lessons to be learned. As we were entering the field, Stanley Prusner's lab was going through this process of small molecule screening against prion-infected mouse cells. This was everyone's best idea at the time. But prion infection is hard to achieve in cells, so you take what you can get. And what they could get was mouse neuroblastoma cells that were stably prion infected. They screened somewhere on the order of like hundreds of thousands of small molecules and read out ability to reduce protease-resistant prion protein in the cells and came up with some leads, actually did a lot of medicinal chemistry, like much more than the average academic lab would be able to do in order to make these drug-like molecules blood-brain barrier permeable, put them into mice. And in prion-infected mice, mice infected with mouse prions, saw a beautiful survival benefit. At the time, the best survival benefit the field had ever seen. Then they put those molecules into human prion-infected, humanized mice, and they saw nothing. And this was like the very hard-won lesson that a prion is not a prion is not a prion. There's actually different structures, probably a cloud of different structures in every case, and we don't know still the mechanism of action of those small molecules, but whatever it was probably specific to certain conformations within that strain. So we look back now, I look at that, and I think we're so lucky to know that. I mean, hard-won, painful information, but I now am not going to put any eggs in the basket of targeting prions themselves, let alone neurotoxicity. We don't know anything about neurotoxicity. So I do think, to your question about why haven't we moved the needle, to move the needle, we need to be focused on the right target. And we tend to focus on what is spectacular, right? Prions are spectacular. They're so weird. There have been like 3 Nobel Prizes. Like, they're the reason our little disease is famous. That's the thing people went at for some number of years. But now we have both the maturity as a field and the technologies coming online to look upstream at the RNA, at the gene. I think these are actually going to be our more tractable targets. And it just took time for all of us to understand that. But also, we should reform academia. But you had a second question, which was?
So what could we do as a scientific community?
Yeah, it was what could you do? So I think like this is a conversation I would love to have with you guys over the next day and a half. I'll be here until like tomorrow afternoon. But if you see in everything that I shared a way to help or a way that we could brainstorm together, I hope you'll come find me. Because I have some ideas, but it is a conversation. And I think a lot of what we can do, right, is just to think about. I feel stuck in some ways in between and of one world where, yeah, there's spectacular examples of moving really fast. I will say that most of those examples are spectacular examples of moving really fast, but it was already too late, right? Both are true. And then there's sort of like commercial development, common disease world. And for a modestly rare disease, we're sort of being lumped over here, right? And in a lot of different ways. Like I look at Eric bent over his desk right now, and the burden on his shoulders right now is not very different from the burden on a 200-person biotech or a 2,000-person pharma company. I don't think that's the system we would have designed according to first principles. I don't think people would tell us, well, just go home and give up. Like, I just think we end up with whatever system kind of crops up and evolves, and then we are in it, understanding that that system doesn't really make sense for all use cases. So figuring out, how to make what we're trying to do a little bit more doable, I think we're not the only use case. And part of our goal is to publish everything. Here's another superpower, right? We're not traditional sponsors. We can just publish it all, almost in real time. I want every single aspect of our IND-enabling studies in the supplement of the paper. Because even figuring out what do we have to do, like even just assembling the checklist, took ages. Not to mention, you're trying to write an IND, and like imagine writing an academic paper without ever having read an academic paper. That's what the exercise feels like. No one shares their INDs. So like, what even is it? Like, you know, maybe you find some, like 2 or 3 academics who are like, I've done that before, like these are what the sections look like. But Eric was saying to me yesterday, I'm certain there are whole chunks of this thing that I just don't know about and that we don't have yet because nobody told me, because there's no template, because this is just a thing that we expect people to know because they have hired $400 an hour consultants. And we have some of those, right? Nevertheless, I think there's so much received wisdom in how this stuff is done, and so much of it you can't derive from first principles. Like you can't derive it from what makes sense. What kind of information might be useful in this kind of submission, right? You just have to like know the sort of like bespoke, inherited art of doing this stuff. You just have to like be in the club. And that's where like biotechs are because they hire all those people, and that's where pharma is, right? You just hire all that intelligence. You just hire people who've done it before. That's our only solution for any of this, and we can't. So yeah, I would love to talk to you guys about like, what do we do besides just like keep getting up every day and like throwing ourselves at this, you know?
Thank you. Good luck.
Thank you.
Great. I think we have time for 2 more questions, likely. So, Dean, please.
Yeah, thanks, Kim. A remarkable talk, a remarkable subject, and you've revised the word transformational for me. I'm Dean Smith. I'm with Health Canada, and believe me, in the government world, the word transformation has been horribly abused. But you've given it new life, so thank you. I want to thank Andrew for his comment and question and your answer. And I want to pick up on that with actually a suggestion. I'm in vaccines, but it seems to me from a distance that at the FDA, wherever your struggles have been, FDA's Gene and Cell Therapy Group is one of the more innovative on the planet. And if you can get to the right people with your story, which I'm sure you never have the opportunity to give as you've given to us today, that's not a typical regulatory exchange, but that's what you actually need to do, and it seems to be your superpower to do. So maybe with the support of CASSS, this presentation you gave today could be made available for you to target to who you feel might be useful. And I'm happy to talk to you about some specific targets that might be useful to you after. But I think if you get this story as presented to us to the right places and people, whether they be industry or FDA or other agencies, you will be very successful. And I want to wish you well with that.
Thank you so much. I love that. I would love to follow up with you about that. I feel very little obligation to be typical.
Good.
So, for what it's worth.
Again, is a superpower.
Great.
Hi, Paul Kerwin from Amgen. So you mentioned you mentioned with your adaptive trial designs to try to avoid randomizing folks in your disease population to death, right? I think the words you used. I was going to make a recommendation just for an idea, but I know that costs are, sensitive for your endeavor. But there is a lot of talk about, in this age of artificial intelligence, of using digital twins in clinical trials and things like this. And so I wondered if there's a way you could leverage that, or if you've talked to people about leveraging that concept to help avoid, randomizing folks in a way you don't want to, right?
Yeah, I love that idea, and I'm going to tell you a really sad story, but I hope that we can turn it around and make it a happy story. But I think any sort of synthetic placebo control arm is going to rely, whether we use AI to further model it out and expand what we have, it's going to rely on some nucleus of natural history data collected by clinicians around the world. And we have been on the warpath trying to get the old guard, who've been doing this for a long time, who've seen a lot of patients, who have a lot of data, to share it in a way that we can then make available to sponsors responsibly, right? Like, let's de-identify it, let's do the things, but also let's agree that the highest use of this data is to make it into one harmonized, interpretable data set that is shared with sponsors and anyone else who wants to help with this disease. And This reasonable, I think, pretty reasonable proposal has been rejected more times than it's been accepted. And there's all sorts of straw man reasons. But I think if I had to boil them down, one would be, hey man, I spent my career building up this data set. This is a publication where I'm the last author and my postdoc is the first author, period. Concern number two is, like, you know, and people will say, oh, like, patient privacy. Okay, like, you can say that anytime we're talking about sharing data for any purpose. You can kind of wave the patient privacy flag and be like, no, I don't want to. But that's me you're talking about, and I'm asking you to do it. So I think we really abuse the patient privacy thing. I'm not saying we shouldn't be responsible, but if you ask those people, hey, what do you want us to do with this data? Should we literally sit on it? Do you want it to be some like dusty publication that's read by 5 people? Or would you like it to be applied to drug development? I think we all know what the answer is going to be. The final thing is, and this one is like hilarious, is that some people say, oh, pharma's interested? Cool. I'll share the data with them when they give me $500,000. To which I say, folks, right? And these are academic. They're like wherever. They're off wherever. I'm in Kendall Square, right? And I think there's just such a fundamental lack of appreciation for how we are situated in the drug development world and how easy it is, right? We are right on the margin of are we commercially viable or not. You make it 1 iota harder for a sponsor, it is the easiest thing in the world for them to walk away from our rare disease. But like sponsors in the room, you shouldn't. Anyone who's interested in prion disease, you shouldn't walk away. But I think academics don't know that. They don't know that. They're speaking a really, really different language. So this is a tragedy that I'm up against every day. And honestly, like, to survive, I have to have a threshold where, yes, like, you actually have the largest data set in the world, but you're dead to me. Like, I just need to focus on the people who I can work with to try to get this done. I would love for us to have the tools to do synthetic placebo control.
Thanks.
Great, thank you. So I'm afraid we're going to have to end some of the questions. We did receive a few more questions online, and there may be more questions that hopefully folks will have the opportunity to speak with you over the next day and a half.
I hope so too.
But I can share some of the questions that arrived offline and try to provide that. But, you know, my tremendous thanks. And I was wondering if you could close on something that just resonated so strongly with me from your TED Talk. And, you know, I think I wrote this to you in the email. I feel like you found us, you know, and there's some purpose behind that. But, you know, the closing line of your talk in terms of a nod towards poetry. So I was wondering if you could just share that idea to kind of close that out, please.
Oh, sure. So what did I say? I think what I said was that we have a tendency to think about our lives, right? And I said this specifically in relation to, Eric and I made the choice to have kids through IVF PGD. So nothing is, you know, bulletproof in biology, but we have a reasonable confidence that these kids are mutation negative, which I think is kind of the ultimate form of prevention. And like, again, a gift of the moment that we were able to do that. But you kind of see as a young parent, right, the way other young parents talk about milestones in the future and like X and Y, and the kids are going to do this, and we're saving for college, and at certain ages, certain things happen. And honestly, like, I hear this, like my sister-in-law was saying the other day, oh, like in 6 years, right, when Carmen graduates from high school, maybe we'll go like live in Iceland for 6 months. I don't know. She had like some wacky plan. And I was like, that must be so interesting to be a species that plans ahead like that. Because I don't take for granted for a second. I mean, don't get me wrong, I would love to be right here in 6 years talking to you guys again. I don't take it for granted for a second. So I think that's what I was getting at, was that we had this tendency to sort of like look at this sort of narrative timeline and think of our lives as this sort of like linear string of things that are going to happen. And I think that is a luxury, right, that not all of us can afford, myself in particular, but I also think that it is a luxury none of us can afford. And this is what I think is actually very interesting about my position in the world, is that it is not unique. It is not actually very unique. We all have a tremendous amount of uncertainty, guiding the parameters of our lives. And how much we engage that on a day-to-day basis varies. But I think when I said poetry, I think what I meant was not only engaging life on this sort of linear narrative level, but having that sense of, of deep time, which in some ways is all we get, right? The rest we make up. But anyway.
Beautiful. Thank you. Please join me in thanking Sonia.